Hello, my name is Monique Christian. My project was to study asthma and allergy and to look at the role of microbiota in asthma and allergy. In this particular study, we focused on the role of microbiota on airway resistance, which is a feature of asthma. The most common type of asthma occurs when a harmless substance or an allergen, such as a house dust mite, can cause an immune response in the airway. The continued exposure to the allergen, the process is repeated and becomes more chronic and more severe. The image on the right illustrates that process. Are all the microbes, both cellular organisms and viruses, that live on or in a location. Our focus is on the human microbiota. The number and variety of microbes are greatest in the GI tract. The image on the right shows that reduced types of specific gut microbial diversity during infancy is associated with an increased risk of allergic manifestation in children. Mast cells are permanently anchored in tissues. They release many chemicals that participate in allergic inflammation and allergies by degranulating. The image on the right displays the many receptors that mast cells release in the airway smooth muscle after allergen exposure. The experimental model contained four types of experimental groups. Microbiota were taken from one group of infants with eczema, identifying them as having potential allergic sensitization with an enhanced risk to develop asthma. The second group was from infants that did not have eczema, making them less likely to develop asthma. The third was from adult humans, which were from apparently healthy young adult humans with unknown allergy statuses. And lastly, from mice with their microbiota undisturbed. Each group contained 10 mice, and the humanized microbiota was transferred as a fecal transplant to germ-free mice. The treatment protocol had a control variable, PBS, and an independent variable, the house dust mite allergen, because it is the most common allergen people are allergic to. The image on the bottom displays the HDM exposure protocol over a span of 2 weeks with the methylcholine, a bronchoconstrictor. This graph is the result of the preliminary experiment. On the top right-hand corner are the four microbiota types. As it shows, infant B had the highest response level than the other microbiota types, and the mouse had the lowest response level than all the others. The preliminary conclusions to date shows that mice with the three humanized microbiota had increased AHR compared to the mice with their microbiota undisturbed. However, the inflammatory responses were similar in all four microbiota types after exposure to the HDM allergen. The hypothesis I tested was that the increased AHR in the mice that received the HDM allergen is dependent on mast cell mechanisms. My methods consisted of three steps. First, to develop a literature search strategy for mast cell and mast cell receptors that are associated with altered lung function after allergen exposure. The second was to identify those mechanisms that can link mast cell activity to AHR in mice with humanized microbiota. And lastly, to create a correlation analysis between mast cell protease in the airway and lung function. You can see the literature search strategy I used here. With the identified mechanisms that can link mast cell activity to increased AHR in mice with humanized microbiota, cluster of differentiation or CD200 and neurotrophin 4-5 or NT4, further work is needed to determine the role of mast cells in allergen-induced responses in the airway. The first plot from our results showed that there was a positive correlation in lung resistance in mice with adult human microbiota after allergen exposure. This plot shows a negative correlation in lung resistance in mice with their undisturbed microbiota after exposure to the control. This plot shows a negative correlation in compliance in mice with adult human microbiota after allergen exposure. And this last plot shows a positive correlation in compliance in mice with infant A microbiota and mice with their undisturbed microbiota after exposure to the control. Our conclusion suggests that we may accept the given hypothesis. I would like to acknowledge and thank the 2020 Brush Program at Michigan State University and all my mentors for making this possible.